Dear children, welcome to online classes of biology. So, in the previous class, we have seen that how glucose is getting oxidized, during different process like glycolysis and also in Krebs cycle. Stepwise oxidation of food takes place during Krebs cycle and during the glycolysis. And we have seen that ATP produced in some places, and we also have seen that NADH, isn't it? NADH plus H plus, and in some other places FADH2 is formed. FADH2. So these will produce ATP, or this has to be oxidized further. Then only what can be produced? ATP can be produced. Understood? So this process, that is the oxidation, further oxidation of NADH, and FADH2, that takes place in another what process, that is ETS. So let's start today. ETS, Electron Transport System and Oxidative Phosphorylation. Understood? So, what is the role, or what is the importance of this? These processes are very much necessary to release and utilize the energy stored in NADH2 and FADH2. So how it is possible? When they are oxidized through the electron transport system. And then what happened? The electrons oxidized means what it will release electrons, isn't it? When electrons are removed, that is said as what? The oxidation. And the removed electrons, finally it will be sending to oxygen molecule, and that results in the formation of water. So we have studied that during the aerobic respiration, glucose combines with oxygen and produces what? Carbon dioxide and water. So till now we have not seen any production of water or the utilization of oxygen, isn't it? In Krebs cycle and also in glycolysis we have not studied. So the use of oxygen comes here only, where in ETS. Because the electrons which are produced due to the oxidation of NADH plus H plus and FADH2, they are released, or they are accepted by the oxygen, and that results in the formation of water. So what is electron transport system? The metabolic pathway through which the electron passes from one carrier to another is known as what? Electron transport system. So here what is happening? The transfer of electrons from one carrier to another carrier is known as what? Electron transport system. And where this electron transport system are present? They are present in the inner mitochondrial complex. You can see that in the inner mitochondrial complexes, they are arranged as different complexes 1, 2, 3, 4, and 5. 5 is the actually the ATP synthase, synthase enzyme. Likewise in different complexes, they are protein molecules, protein containing iron, and some are there which are having copper. Understood? So like this, in the inner mitochondrial inner mitochondrial membrane, you have learned now, mitochondria is having outer membrane and inner membrane, and the inner membrane is folded into different what structures called cristae. So in the inner membrane only you can see what? The complexes arranged, and through this complexes only the electron passes during the ETS. So here you can see this is the matrix, and this is the membrane, and this is the matrix, and this is the inner membrane space. Is it clear? So through this complexes. Okay, so ETS is arranged where? In the membrane. And the NADH2 and FADH, okay, they pass their, okay, electrons to this complexes. Okay, from one to another, the electrons will be passed, and that will be simultaneously coupled with the synthesis of ATP. Understood? So electrons from NADH produced in the mitochondrial matrix, isn't it? In the matrix only this Krebs cycle occurs. Okay, so this will be passed to where? To the various complexes, okay, molecules which are arranged in the membrane. Understood? So the first complex, that is NADH dehydrogenase. Okay, and then what happened? Ubiquinone, another molecule, okay, receives reducing equivalents, okay, that is electrons from FADH, okay, and the reduced ubiquinol that will be getting, means they are arranged according to their what oxidation reduction potentials. Okay, so when one molecule is getting oxidized, understood, by giving its electron to next one. Okay, when electron gets, they will get reduced, and when they give electrons to the next one, they will get oxidized. So like this complexes can get oxidized and reduced, okay, alternatively. When electrons comes, they will get reduced, and when electrons goes to the next one, that will get oxidized. Okay, so the NADH2 gives its electron to what? NADH dehydrogenase. Okay, that is complex 1, and the electrons are then transferred to ubiquinone. Okay, that is also located within the membrane only. Ubiquinone then what can also take the electrons from FADH2. Okay, so like this, when the reduced ubiquinone, okay, they will give its electron to cytochrome C via cytochrome BC complex. So complex 3 contains what? Cytochrome C and cytochrome BC1 complex. Okay, so ubiquinone give its electron to complex 3. Understood? Now cytochrome C is a small protein attached to the outer surface of the inner membrane, and this is the only mobile carrier. Okay, so mobile means it can move between complex 3 and 4. Understood? And complex 4, that refers to cytochrome C oxidase and complex containing what cytochromes A and A3, okay, and 2 copper centers. So that is what complex 4. Understood? So like this, you can see the electrons which are coming from NADH2 will be passing from one complex to another. Understood? And finally, what happened? That results in the ATP synthesis. Okay? So, in this way, when electrons pass from one carrier to another, okay, through complex 1 to 4, what happens? You can see that they are also coupled with ATP synthase. ATP synthase are the what enzyme actually, isn't it? Which are used for the production of ATP from ADP and inorganic phosphate. Okay, now then we have learned that NADP is equivalent to produce 3 ATPs, and FADH2 is able to produce 2 ATPs, isn't it? So that on this basis, means which compound is giving what or getting oxidized. Okay, the on that basis the number of ATP also differs. For example, FADH2 can produce only 2 molecules of ATP. Okay, and NADH2 can produce 3 molecules of ATP. Understood? So although the aerobic process of respiration takes place only in the presence of oxygen, the role of oxygen is actually what present here. Understood? In the terminal stage of the process, that is to accept the electron finally. Who is needed? Oxygen is needed, so that what will happen? Oxygen will get converted to water. Okay, so this is the, and this is very important also. Without oxygen, ETS cannot run. Understood? Even though the role is in the last stage, but it is very important. Okay, without oxygen, the ETS cannot be done. Okay, if ETS cannot is not there, what will happen? NADH2 and FADH2 cannot be oxidized. If it is not oxidized, then what happen? Electron means the ATPs cannot be produced. So finally, the ATP production, okay, happens where? In the ETS. Understood? So like this, what, oxygen acts as the final hydrogen acceptor. Understood? Now, in we have learned about photophosphorylation, isn't it? Where what happened? Light energy is used for the production of proton gradient. Okay, we learned the chemiosmotic hypothesis, isn't it? Means we learned that in the membrane, the thylakoid membrane, means inside the membrane and outside the membrane, a proton gradient is created. And when this proton gradient is broken, okay, that is associated with what? ATP synthesis. In case of what? Photophosphorylation. In the last chapter, we have learned, okay, in the photosynthesis chapter. And here in respiration, okay, the oxidation, that energy of the oxidation reduction, okay, is used for creating the proton gradient. And it is what, the this is the reason it is also known as what? Oxidative phosphorylation. That is for the production of this proton gradient. Here also proton gradient is created, but the energy for this comes from what? The oxidation reduction of what? Oxidation reduction, the energy of oxidation reduction. Okay, as the electrons pass from complex 1 to 4, that is used for what? Creating the proton gradient. And it is this is the reason it is not also known as what? Oxidative phosphorylation. Okay, now how chemiosmotic hypothesis explained what ATP synthesis? Do you remember? Okay, we learned that the energy released during the electron transport system is used in what? Synthesizing ATP with the help of what? ATP synthase. So ATP synthase is actually what? The complex 4, which is also located in the inner mitochondrial membrane. Okay, and this is having two parts, just like the ATP synthase of photosynthesis. Okay, here what happened? It is having two components, F1 and F0. Okay, the F1 is the headpiece. Okay, and it is a peripheral membrane, peripheral protein. Okay, we have learned no peripheral and integral proteins. Okay, so peripheral proteins means they are placed in the super what? Visual side, isn't it? In the side. And F0 particle, that is a integral protein. You can see that is spanning in between the membrane. It is well embedded in the membrane. Understood? So the F1 headpiece is a peripheral membrane protein complex, and this is the site for ATP synthesis from ADP and inorganic phosphate. Okay, F0 is what? It's a integral membrane protein complex. Okay, and through which only what? Proton cross the inner membrane. Okay, through this proton comes out. Okay, the proton gradient is first created where? In this inner inner mitochondrial membrane, or inner mitochondrial space between the inner membrane and the outer membrane. Then once the gradient is in the created, then what happened? The breakage of this gradient takes place through this channel. Okay, that is through the F0 particle, and this results in the production of ATP. That is, inorganic phosphate and ADP will get joined and produce what? ATP. Okay, so 2H plus passes through the F0 from the intermembrane space to the matrix down the electrochemical proton gradient. Okay, 1 ATP will be produced for each 2 H plus ions that passes through the F0. How many ATPs will be produced? one ATP will be produced. Is it clear? So in this way, what you can see that the electron transport system, okay, that helps in oxidizing what uh, the NADH2 or FADH2, and along with that they create the H plus, okay, gradient in the inner mitochondrial membrane and the matrix, membrane, not inner mitochondrial space, that is the space between the inner membrane and the outer membrane, and then also towards to the matrix, and this results in the what formation of ATP. So what are ETS? Okay, it's a metabolic pathway through which what electrons get transferred, isn't it, or passes from one carrier to another. Okay, and it is present where in the inner mitochondrial membrane. Okay, and you I have said that they are arranged in different complexes, complex 1 2 3 and 4, and finally the 5 is actually ATP synthase. So the ETS is always associated with what, the ATP synthase. Okay, these are the what proteins. Okay, so like this, what the process of electron transport system, okay, is taking place, and it results in the formation of water and ATPs. Okay, this much is very clear. I hope we will continue in the next class.